the window that is this. Okay, let's make sure that this is working. Can you guys see the screen where it says how populations evolve?
Yes.
Yeah. Excellent, great. Okay, so we are going to be looking at how to use math to prove evolution. And again, I feel like I'm teaching to an empty room because you guys are over there on that computer screen and there's nobody in here. This is really weird for me. So let me know if you guys have any questions. This right here is the Hardy-Weinberg theorem. It's of two formulas, p squared plus 2pq plus q squared equals 1, and then p plus q equals 1, okay? It looks complicated. It's really not. It's just a quadratic formula. It's super easy once you do a little bit of practice with this. And this is another one of those things with math that, that you know, we have to kind of cover here in AP Bio. So let's see kind of where we get this and what we're talking about here. All right, so the first thing we got to do is think of a population, because this is all about population evolution. There's another name for it. It's called microevolution, because it means like really small evolution. I guess I can look in this one right here, although I can't see myself, so it's kind of weird. It means really small level evolution, and we'll talk about how those two things are different, micro versus macroevolution. But in this one, we're looking at measuring allele frequency in a population. We're going to figure out what that means. So I'm going to put some numbers up here. This is an island. And this island it should have 10 organisms. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8, 9. Looks like I have 9 organisms instead of 10. Maybe we add another one in here just to make it a little even number there. So we'll put a big A, little a. Okay. There's 2 words here that you have to be able to distinguish between, and that would be genotype and allele. Okay, this sounds more like genetics at this point, and it really kind of is. But genetics and evolution are just, you know, they're really so closely related. Okay, so when we talk about genotypes, the genotype is the combination of genes that we get from our parents. So is that going to be 1 letter or 2? 1 letter or 2. Good. I saw Seema. I'm looking at people's pictures here. That is 2. So the genotype possible options that we have here are big A, big A, big A, or little a, little a. Those are the genotypes, okay? We can call them homozygous dominant for this one, heterozygous, and homozygous recessive right here. The alleles are just going to be the individual type of gene, the dominant and the recessive allele right here, okay? And when you talk about evolution, it actually becomes much more important to think about the allele, and we'll see why with the Hardy-Weinberg theory in just a minute. The other thing here is frequency. Okay, so we need to think about a frequency, which is kind of like saying a percent, but you don't multiply by 100. And so we want to think, well, what is my homozygous dominant frequency? So can you guys just unmute yourself if you want? How many out of how many are homozygous dominants in my island population here?
2 are homozygous dominant.
So 2 out of 10, because I added that extra one in there, which equals 0.2. My homozygous dominant frequency is 0.2. What's my, is that right? I only saw 1, 2, yeah, I did, okay. Yeah, you guys are right too. Okay, what's my heterozygous frequency?
0.6.
0.6, yeah. So let's make sure we're not thinking 60% because you can't really do math with percents. So we just have to think about the decimal version here. And that means my homozygous recessive frequency would also be 0.2, and that is going to equal 1 because there's no other organisms on the island. They have to equal 1 right there. Well, okay, now let's think about the allele frequencies, right? Because each individual has 2 alleles. So all of a sudden, the bottom of the fraction, the denominator, one of the others, I don't know, it no longer is 10, it is 20. And so now we can go through and count, okay, how many dominant alleles are there in all of the different individuals? I got a number?
There's 10.
There's 10. So 10 right there, which means this has to be 10 because there's nothing else. So we would then say this is 0.5 and this is 0.5. Okay? So now we have two things. We have genotype frequencies and we have allele frequencies. If we walked to this island, would we be able to figure these numbers out? Would we be able to look at the individuals and know which ones were what? If this was like eye color or something like that as a trait.
Well, we could know which one had dominant versus recessive because the dominant is always going to show. So we couldn't tell if they were homozygous dominant or heterozygous necessarily, but we could tell whether or not they had a dominant allele.
You can't tell between those two right there. So that means that you can never know those numbers because of like the inability to tell, right? You can't figure out which ones are homozygous dominant or heterozygous unless you have some sort of DNA test that'll actually tell you that. But we can use math to figure out what they're most likely going to be. Then we get the whole idea of evolution. What Hardy and Weinberg, it was two guys that did this, and this was way back in like the 40s or something like that. They said that evolution is actually defined as changing allele frequencies. Like if we let this population just grow and reproduce and die, grow, reproduce and die, grow, reproduce and die, and then we come back after 10 generations or after one generation, it doesn't matter. The top right here, the genotype frequencies, they might have changed. These up here, they might be different, but that doesn't matter. All that matters is the allele frequencies. If the frequency of the alleles in the population hasn't changed, then we would say no, the population did not evolve. So like our new definition for evolution is changing allele frequencies in a population. The way the math works is this. Okay, so I don't know the genotypes like y'all just said, and let's imagine this population right here, or not this population, this genetic cross, a heterozygous cross, okay, big A, little a cross to big A, little a. And we're gonna switch some numbers out. Instead of representing the alleles of big A and little a, we're gonna let the frequency of big A be a lowercase p, and the frequency of little a be a lowercase q. So then all of a sudden we can put P, Q, P, Q right there, and we could fill that in with numbers if we wanted to. We really don't need to do that. We're just showing where the formula actually comes from. And then the combination up here, what are we going to put in the top left square of the Punnett square? If this is the frequency of P, and this is the frequency of P.
PP.
Okay, but let's rewrite that as P squared, okay? Because it's P times P, which, you know, we'll just rewrite that as P squared. So what's going to go down here in the bottom right?
Q squared.
Q squared. What's going to go in the top right?
PQ.
P, Q, and then P, Q right here. So if you have the allele frequencies, then you can start filling this in, and all of these in here, like those are the only possible combinations you get. And so you can take that and say everything has to add up, and you get P squared plus 2PQ. We just combined those right there. Okay. Plus Q squared equals 1. Okay, because you can't have it be anything else. The other formula we can get is P plus Q equals 1. So this is the genotype frequency, and this one is for the allele frequency. And so what the formula actually means, P squared plus 2PQ plus Q squared equals 1, and P plus Q equals 1, this is the frequency of big A, the frequency of big A, and the frequency of little a, the frequency of big A, the frequency of little a. And that means we can do some math, because if we know some of this, like if we happen to know, maybe, I'm going to try to switch to a different color here. If we know this, because we go to that island and we count up all the whatever animals with blue eyes, if we know this number right here, the frequency of individuals that are homozygous recessive, can I then figure out the frequency of individuals that are heterozygous?
The genotype one can.
But I can go from this one to that, because all I got to do is take the square root of that frequency of q squared. Because q squared, take the square root, and I'm going to get q. And then I can figure out p, and then I can put those two into that formula, 2 times p times q. And I can figure out how many of these animals are heterozygous in this population because I knew the homozygous recessive ones. And that math becomes really powerful because you can figure out populations where you don't have like a genetics test. Now this is one thing, it's one thing to like talk about this. We can talk about this like all day, but we need to find like a practice problem. Okay, so this island has 15 individuals. Let me see if I have another one. I don't. I'll have to switch over to the PowerPoint to do another one here. I'm going to do this at the same time as you guys. Okay, so what I want you to do is look at this island. You should be able to read it. Can y'all read the letters on the island? Everything is clear enough?
Yeah.
15. You can't read it? Is it too small?
No, well, I can. I don't know about everybody else, but.
Okay, so what I want y'all to do is come up with 5 numbers. From this island, can you tell me the frequency of homozygous dominant, the frequency of heterozygous, the frequency of homozygous recessive, the frequency of the dominant allele, and the frequency of the recessive allele? I'm going to do it too because I just, this is, I don't have the numbers yet. I need to just pick. Okay, I got my numbers. I'll give you a second. I'm going to try to figure out how to do attendance here. Brittany, is Tiffany with you?
Yes.
All right.
So do we not fill out that attendance sheet anymore?
That's a good question. Yes. I just got y'all right here, but because, people might be doing different things. I do want y'all to do that. Now, if you're here right now, don't worry about it, but let's try to make a habit every time we start class, like we just go in and do the attendance sheet. But if you haven't done it today, don't worry, because I just marked y'all present for today. So yeah, that's a good question. Okay, it looks like I made really super easy math here when I made this one. I think I made this one last year just as like another example. Anybody know what q squared is?
.3
Five?
repeated.
Ooh, I got it, 0.3, yes, because over here, this one is q squared, and it would be 5 out of 15 or 0.33, and that'll be enough right there. Okay, so remember, that one is q squared, p squared is the same thing, and 2pq is the same thing, so we get 0.3, 0.33, and we'll add those just to make sure, okay? Okay, what's q?
0.5.
0.5, okay. So 0.5 and then 0.5, because that would be 15 and that would be 15. I'm looking for my calculator. I got it, okay. So we were able to do this without having to use the Hardy-Weinberg formula, only because I put the genotypes of all these individuals up here. But most likely we wouldn't have known this. Most likely the only thing we would have known is that right there and that right there. Okay, we would have known that 0.33 were expressing the recessive phenotype and 0.66 were expressing the dominant phenotype. And we would have had no idea over here which one was which, because we wouldn't be able to tell. Like, there's no way you can do that unless you start using the math. So the square root of 0.33 is not exactly this. It's actually 0.57, okay? And so the point of that is to say, like, if you can count individuals, like if you can see them, then do that. Just count from this, right? Because I've seen College Board questions on AP tests that have you just count. Oh, it's 15 out of 30, or oh, it's 5 out of 15. But if you can't count, that's when you do the Hardy-Weinberg formula, and that's what I'm going to switch over to for the PowerPoint here in just a second, okay? All right. Usually, this is something that we get lots of practice on. Like, we'll be doing practice all week long on this kind of stuff. It can take a little bit of time because it's usually a little bit new, like working with individuals and frequencies and math with evolution and that kind of stuff. But remember, what if we were Hardy and Weinberg? What would we predict would happen, or what would it take for us to say that evolution had occurred? Next generation, what would it take?
A change in frequency?
Which frequency?
Both?
Allele frequency.
Allele frequency, yeah.
Allele. It would have to be a change in the allele frequencies, because there is some math that you can do where, like, if you take, and I forget, this is really hard to just do by hand, but if this one goes, I think, down by 2 to 3, and this one goes up by 1, and this one goes up by 1, I don't think that changes, or no, that changes those. Like, there's ways that you can do the math that these don't change, but it's the allele frequencies that really matter. All right, let me see if I can figure out how to switch over to PowerPoint, because everything is really confusing. Okay, I'm gonna stop presenting. I'm gonna go over here to this. Okay, I gotta present this too. I actually feel like a teacher again. I'm in a classroom, I'm teaching, this is fun. All right, so let's see if this, it looks like you guys can see this. Where'd it all go? Can y'all still see it?
No.
Present now. Present the window. Is it back?
Yes.
All right, excellent. So we're still doing the same thing, but we're just going to look at it in a couple of different ways here. And I'll post this. I may have already posted this online. I don't know if I included this one. I can certainly add it. But when we talk about, I want to look at this one first. Okay, so I always like to start this kind of, this topic with what we call microevolution, what we're just talking about. That's like really, like, microevolution is really easy to prove. Like it's just looking at populations from one generation to the next, and you can just do some genetics tests and you can figure that out. We're going to finish up this unit with macroevolution. That's things that happen over long periods of time, and like, you know, where did humans come from? When did we first show up? Can we even prove that? Was it even at one point in time? That's really big scale things, and so that's micro versus macro. And to connect those two, we have this concept called speciation, or the origin of species. Like, how do you go from one species to two? And so I like to start with small, and then speciation, and then macroevolution at the end. So we will go in that order. And we say, like, this whole idea of population genetics or microevolution, it's the study of allele frequencies. So that's what we're talking about right now. All right, let's see. I had a practice problem here. Okay, we're gonna keep going. All right, so I like this one because, like, it's kind of a good analogy. Analogies really help us understand things, right? If I took a deck of cards and they were in a certain order, right? And how many hearts would there be? Do you guys know your numbers on your decks of cards? How many hearts? You guys not play cards? We've already had our trip to Vegas for chi-squared, so there's 13 hearts, 13 spades, 13 diamonds, 13 clubs, and they would be in a certain order, in a certain pattern inside that deck, right? And then if I shuffle it, I'm going to change the pattern, but does that shuffle change the number of hearts? It doesn't. Good. I see y'all shaking your head. It doesn't. It changes the order and the pattern, but it doesn't change them. And so what Hardy and Weinberg said is that the frequency of alleles and genotypes in a population's gene pool remain constant over generations unless acted upon by an outside force. What they meant by outside force were the 5 things that can make a population evolve. Let's look at those 5 things, and then I will show you a practice problem. So, all right, so they said to have a non-evolving population, right? If you have this population and you're going to say that population is not evolving, it has to be large. Okay, being large removes the effect of sample size, and it's the same thing we talked about with chi-squared. You flip a coin twice, and it's not uncommon to get 2 heads in a row. But if you flip a coin 200 times, it would be like infinitesimally small chance of getting 200 heads in a row. So the large population size is going to remove sampling issues. You can't have migration because migration could bring in a new allele that you didn't have before, and all of a sudden the allele frequencies have changed. If you have a population that is 100% like P, like dominant, and then in migrates this recessive individual, then you've changed the allele frequencies due to migration. You can't have mutations because mutations can change an allele from one to another. You have to have random matings because random matings will make sure that, like, if all of the, like, if there was some selection against the homozygous recessive individuals because they're not like seen as like mating worthy or something like that, then that's going to lower Q, and you have to get rid of natural selection. Okay, so this is a non-evolving population. Does this exist? Is this something that can happen in the world, in reality? I see Seema shaking her head. No, she's entirely right. Okay, you can't have this. You can have a population that is large. You can maybe on an island have a population that has no migration. You can probably have a population where random matings are mostly occurring. And I don't know, I mean, you really can't get rid of natural selection, but you can definitely not get rid of mutations. So like in a zoo, a zoo is going to get rid of natural selection, right? Because like we don't let the lions eat the slow zebras. They're put in their pen, right? And in a zoo, you know, there is no migration. But in a zoo, there's a small population size. In a zoo, you can't stop mutations from happening. And so this the whole idea of like Hardy-Weinberg, it's just a thought experiment to say, this is what it takes for a population to not evolve. And if this is what it takes for a population to not evolve, then that doesn't exist. Every single population is evolving all the time. All right, I'm gonna leave the mechanisms of evolution for probably tomorrow, because I wanna go over this one right here. Let me make that bigger. I don't know what just happened. I tried to present, and let's close that. Am I still sharing? Do y'all still see the screen?
Yes.
Okay, but it's not on my Promethean board here. Now I don't know what to do. I'm gonna try turning it off. It'll take a second. Are y'all ready to come back to school? Good.
Yes.
Yeah, definitely. I'm working on my seating charts and stuff like that. I don't know. Can y'all see? Let me see if this will turn. You see my personal like sneeze screens? So that way, if somebody sneezes or coughs, then you know we're all safe, I guess. And I've got all these extra masks, and there's only like 14 tables and chairs in here, and I don't know. It's weird, but at least we're back here. So. Okay, let me see if that turns it back on. Slideshow. Don't use presenter view. Okay, I'm just gonna do this and see. Can y'all see that? It looks like you can, because I can see it on my other one. So, all right. If you can see that, then I've got the two formulas here. By the way, they are given to you on the formula chart, which I don't know how the formula chart worked on the computers. I never asked the students last year. I assume there was just a button to pop up the formula chart. But they are given to you. It doesn't tell you what everything means. Like, it doesn't tell you that P equals the frequency of the dominant allele and Q equals the frequency of the recessive allele. It doesn't tell you that P squared is the frequency of homozygous dominant genotypes. But they are at least there, and we usually do enough practice that it's not really a problem. And this is a really standard kind of practice problem for a Hardy-Weinberg question. In a population with two alleles for a particular locus, locus is one of those complex words that they like to use because it sounds fancy. It just means at a particular place. It's the same root word as location. A place on DNA is all it means. So big B and little b. The allele frequency of big B is 0.7. What would be the frequency of heterozygotes if the population were in Hardy-Weinberg equilibrium? And they always finish with that because they want you to think, oh, if it's not changing. Okay, so we know one number, and we're trying to figure out another number. Can you guys figure out that? And I'm gonna, y'all think about that for a second while I try to get this screen working again. So that may have done weird things. All right, there it is. And okay, the way I always think about these, and this is what I always tell people to do, okay? I'm gonna go, did I get it right? Okay. Don't get a pen. pointer options. Yeah. Oh, okay, now it's showing up. I don't know what happened there. P squared plus 2pq plus q squared equals 1, p plus q equals 1. Okay, there are 5 numbers here that are possible to be given or to get. And so I write them down and I say, okay, there's a number there, a number there, a number there, a number there, and a number there. Okay? What am I solving for? Am I solving for p squared, 2pq, or q squared, or p or q? I'm solving for 2pq. So I'm going to put a little star right there. I need to get to that point. I don't care about the other numbers. I might get the other numbers in order to get to that point, but I don't actually care about finding them. I'm just looking for 2pq equals what? Well, I'm given the frequency of capital B is 0.7. Where can I put 0.7 on this?
As P.
0.7, right there. From there, I can figure out everything else. Okay? What's Q?
0.3.
0.3. And maybe I was going to go solve for this one right here for Q squared, and that would be 0.3 times 0.3 because it's just Q squared, and that would be 0.09 right there. But I don't care about that. I don't have to do that. All I have to do is 2 times 0.7 times 0.3. I got no idea what that is.
0.42.
I'm going to trust you on that one. Actually, no, you're right, because it's right there. I see it right there. It's on the answer sheet. So there it is. Okay. There's one other little wrinkle that they might ask. Like, if this was a population of 1,000 individuals, how many would you predict would be heterozygous? It's kind of the same question, and it's kind of a different question, right?
420.
420, because that's a frequency. And so if we have the frequency times the number, then that would be 0.42 times 1,000 equals 420. So even though I can't look at this population and know which ones are heterozygous, I can look at this population and say, you know what, I'm going to expect 420 of them are going to be heterozygous because the math tells me that it is. So, all right, that was good. That was like a real lecture. This was fun instead of me just recording a video in my office at home. Okay, what about an assignment for today? I wanted to start with something simple. And we can all do this kind of together, but we've got a multiple computer mice, everything is weird. I'm gonna discard that. If that one didn't make sense, tomorrow we're gonna do several different practice problems and quite a few of them. So we're gonna keep going with this. And today's lab is actually gonna be easier. It doesn't even include Hardy-Weinberg. We're gonna build up to that one tomorrow. Let's see if I posted it. Let's see there. I don't know if I'm presenting. I'm going to stop presenting for a second. And then I need to go find it here. All right, so I found another virtual lab. And I'm going to present this whole window here. And it's right here. And it's called Population Evolution. And the notes, like I said, are right there. I can't give you guys the active inspire the hand-drawn notes because they just you won't be able to open it up anyway. So but I will post the video here if anybody wants to go back and watch the video, for people that aren't here to watch the video, things like that. But this is the lab that we're going to look at, and it's not very long. It's called a heterozygote advantage, and I wish we could do this lab here where it's a really fun lab to do because we wander around the classroom and we exchange poker chips that represent alleles, and then we kind of bump into each other, and then that represents like, okay, these two are going to make an offspring. We're going to see what kind of alleles they pass on. It's the exact kind of thing that we're not allowed to do because we're like, you know, interacting together and handing things off to one another. So there's no way I can do it. It doesn't matter. But this is going to show us kind of the same thing. So there's a simulation here. and it usually takes a second to like load the program. So if you click on it and it takes a second, that is fine. It'll be right down here. And we can do all kinds of things. We can change the population size from 10 to 1,000. We can change the number of generations from 5 to 100. We can change the allele starting frequency of 0.5, and so we can make the population start out as like all blue because it's red and blue alleles. We can change survival chances. We can change mutations. This sounds like some of those 5 things that would be like in a non-evolving population, right? Mutations, natural selection, the population size, and then what we don't have is non-random matings, but we do have like the beginning stuff right here. So let's see what happens when we run this simulation like this with, let's give it 1,000, and let's just do 50. Okay, it gives us numbers on alleles, and it says phenotype right there, but that really means genotype, because in this case, the heterozygous individual is purple instead of, let's see which one is dominant, instead of red, and so it doesn't really matter. It means the same thing. So we have P, Q, P squared, 2PQ, and Q squared. And we can change all kinds of things here. And the example here is, that we're going to go through is a heterozygote advantage. That is an example where individuals that are heterozygous for maybe a disease have a benefit over individuals that don't have the disease. Has anybody ever heard of something like this before? A heterozygote advantage. It's a super famous biology example.
Like sickle cell?
That's exactly the one that I want you to think. And remember, if it comes up, the sickle cell example, if I can, and we'll talk about this one over and over and over again. Let me see. I want to switch to this and that. There, present a window. Okay, so sickle cell anemia is a disease where, if you haven't, I don't know, I feel like I made a video about it, but I can't remember if that was in anything at all. Doesn't matter. It's a disease where your red blood cells become sickle-shaped because of a mutation. And people that don't have either of these bad alleles don't have sickle cell anemia. People that have both of these alleles do have sickle cell anemia. And people that have, and so let me put that up on the board. So you would have like big S, that would be normal. Big S, that would also be normal. And then little s, that would be sickle cell. Okay, if big S is P and little s is Q, and this one is fatal, although not necessarily at a super early age anymore, but it still is really, really serious. For a long time, it was fatal at an early age, and now we can treat it okay. Would you expect in this case to have P and Q close to 5, or would they be like far apart from one another? Like, which one would be bigger, P or Q?
P.
P, because Q squared is fatal, right? And so all the individuals that end up with this one don't live long enough to pass it on. So we're going to end up with something where like P is equal to 0.99 and then Q is equal to 0.1, right? It's still in the population because the heterozygous individuals can have it, but it's not like super high levels in the population because of the fatal effect of the homozygous recessive ones. But what if we go back? Can I go back? Let's see. Back. What if we go to an area where malaria is present? Do you guys know what malaria is, what causes it, or what it does to a person?
It's the mosquito thing.
It is. What kind? It's passed through mosquitoes, but what kind of organism is it? Like what?
Is it a protist? Or?
It is. It's not a bacteria. It is kingdom Protista. And it lives in someone's blood, in mosquitoes and lots of other apes as well. And it feeds on blood cells, and it goes through a life cycle that involves red blood cells, and so it's really bad for the body, and it's very fatal. It's one of the worst diseases. out there, but it's not everywhere because that mosquito doesn't live everywhere. It tends to be very equatorial on the planet. And the weird thing is that these guys are protected from malaria. Okay, heterozygous individuals are protected from malaria. They cannot get it. They actually can get it, but it's not as serious. And it's something about the sickle red blood cells, and this is where I lose track. I don't know why they are, but it has to do with the life cycle of the protista, and they can't like finish their life cycle in these messed up red blood cells. And what that's going to do is take Q and raise Q and lower P. But if Q is higher because of malaria, what's that going to do to the sickle cell race? If malaria has, it's going to do what?
Did you say what happens to P if malaria is?
If Q is higher, because of malaria, what happens to our rates of sickle cell in those populations?
They would go up, right?
They would go up, and that's exactly what has happened in the course of human history. Anybody whose population, whose like historical population comes from an area where malaria is common, and we can even put like maps up here, which I could do, but I'll wait for that later on. Their populations, African Americans, Southeast Asians, in people who's from India, from Indonesia, where malaria is a really big deal, have super high rates of sickle cell anemia because evolution was protecting them from malaria. So that's what this lab is going to allow us to prove. So let me stop sharing again, and then we can just make sure we work on that. All right, so the lab, share that one. Window, there, share. All right, so the first one you're going to do is the deleterious. That means harmful, recessive. This is going to be just like sickle cell anemia. You're going to set the population size, and then you're going to run and record the final blue and red allele frequencies, and then we'll get the average right there after 5 runs. And then there's an example. And then we want to set the conditions to create a heterozygote advantage. And that's going to set red survival to 0.5. And let me see what that would be. Red survival at 0.5. And I think for the beginning one, we set blue survival to 0.1. And so that's what causes it. So for part 1, the deleterious recessive, we set it to that. And for part 2, the heterozygote advantage, we set it to that. And we're just going to see what that does to the numbers. I think, I mean, it shouldn't take y'all too long. It's a perfect amount of time to kind of finish this up. I think just work on it, and I'll keep this computer open and see if I can help.